Okay, good evening, everybody. It's 7:30, so let's get started. I'm Vivian New. I'm going to be the host tonight. And so tonight we have 4 of our scholarship winners who are going to be talking about their projects, and I want to welcome you to the talk. This is the Santa Clara Valley chapter of the California Native Plant Society. And whoops, what is going on? Before we go into our introduction, I wanted to acknowledge that the work done by the Santa Clara Valley chapter of CNPS lies in the homeland of the Muwekma Ohlone, the Amamutsin tribal band, the Tamian Nation, and the Ramayatush Ohlone, who still live and thrive in this area today. We hope to learn from them and support their work to restore traditional practices and heal from historical trauma. If this is your first talk, we would love to know how you found out about us and where you are. So if you're comfortable with it, please go ahead and share that in chat. And these talks are the work of a team, not just me, but I am your host tonight, and I'm also taking care of the technology in the background. We have a QA moderator who is Judy Fennerty, and she's watching both YouTube and QA and Zoom. And so for anyone who has questions or comments, you can type them in chat, and Judy will be monitoring both and relay the information to our presenters. And we, as I mentioned before, we have 4 of our scholarship winners: Reid Kenny, Lauren Hamm, Alyssa, and I should have asked you how to pronounce your last name, Alyssa, Anzalone, hope I got that right, and Nick Mazzotti tonight. If you are not familiar with the California Native Plant Society, we are a nonprofit environmental organization that was founded in 1965. We have over 10,000 members in 35 chapters that are spread all over California and beyond, not just the state border, but also the country's border, because we also have a chapter in Baja California. Our chapter, the Santa Clara Valley chapter, covers all of Santa Clara County and southern San Mateo County. And CNPS's mission is to save California's native plants and their habitats by bringing together science, education, conservation, and gardening. If you're not currently a member of CNPS, we would love to have you join us. We definitely welcome your support. And once you become a member, you get not just 1, but 2 magazines: Artemisia, which is a scientific journal, and Flora, which has general interest articles about California native plants and their habitats. They're really both quite wonderful magazines and really fun to look at. You'll also receive our Blazing Star chapter newsletter, which gives you information about our activities, such as this talk tonight, field trips, and more. And then you also will receive discounts at participating local nurseries. And of course, the most important thing, you're helping to support the preservation of California's native plant diversity. So if that sounds like something you'd be interested in doing and you're not currently a member, just go to cnps.org/join, or if you have a QR code reader, you could snap that QR code and it'll take you right to that page, and you can sign up online. We have several events coming up at the end of the month. Our photo group has its normal meeting on the 4th Friday of the month. And so if you are interested in photography, if you just enjoy looking at beautiful pictures, please feel free to drop in on that meeting. You can also sign up to share pictures if you're your photographer. We also have a weekly habitat restoration session at Lake Cunningham in San Jose. That is every Saturday from 8 to 10 a.m. You can find out more about that on Meetup, or you can go to our website, cnps-scv.org, and you can sign up for both of those activities. Let's see. Our chapter also has a nursery, which actually it just closed for the summer, but I'm going to go ahead and pitch it anyways. We grow plants year-round, and when we are open, which will happen again toward the end of the summer, you can order plants online and then either arrange to have a time to stop by the nursery and pick them up, or have them delivered if you live between Belmont and San Jose. All the proceeds from anything we sell at the nursery go directly to support our chapter's activities. We're all volunteers, so it's really the way our chapter keeps the lights on and supports activities like this. The other way to find out what's going on besides looking on our website and on Meetup is to get on our chapter news mailing list. I really urge you to do that if you're not currently on it and you're interested in what we do. It's just one message a week. It lets you know what's happening, things like this talk tonight, but also field trips and other activities. So very easy. It's a Google group. You can send email to that address at the bottom of the page, or you can go to cnps-scv.org, and there's information right at the top of the page on how to join that email list. And so just a little bit of housekeeping before we launch into the talks. I would like everyone to make sure your microphones are muted. In fact, I'm just going to go hit the mute all. But if you have questions, as I mentioned before, we have Judy who's monitoring chat on both YouTube and on Zoom. So feel free to ask questions at any point at any talk, and then Judy will relay them to the speakers either at the end of the talk or at the end of the whole presentation, depending on people's preferences. And we do expect to finish by 9 p.m. As I mentioned before, this is both on YouTube and on Zoom. It's recorded on YouTube and it'll be available for viewing immediately after the talk or during the talk and later. So if you want to go back and look at it, or if you want to share it with friends, you can always find it there. So we will be kicking off with Alyssa and Nick, who are from UC Santa Cruz. Their project was, does seedling and planting plus nitrogen addition result in differentiated seed bank composition in restored California coastal grasslands? And Alyssa is a fourth-year undergrad at UCSC. She's studying nitrogen deposition and restoration ecology. And next year, she's transitioning over to aquatic ecology and partaking in the NOAA CCME Graduate Fellowship at CSUMB. Nick is a third-year student at UC Santa Cruz studying environmental studies with an emphasis in concentration and policy. Over the past year, he's been working on his undergraduate senior thesis, and he won our scholarship. So we're very happy to have both Alyssa and Nick, and I'm going to turn it over to you guys now. So I'm going to stop sharing, and it's all yours.
Thank you so much, Vivian. Alyssa, if you want to start sharing. Hi, everyone. My name's Nick Mazzotti. I use he, him, his pronouns. Alyssa, I think you're still muted.
Sorry. Hi, everyone. My name is Alyssa Anzalone, and I use she, her pronouns. Nick and I are currently both undergraduates at UC Santa Cruz, and we are very excited to be sharing our research with you this evening.
Tonight, we'll be presenting our joint senior theses, which discusses California coastal grasslands and how they persist through site seed banks, and how these grasslands are affected by nitrogen addition. So we wanted to begin with a land acknowledgement. We carried out all of our research at UCSC, and we felt it was important to acknowledge that the land in which we conducted our research on was the unceded territory of the Awaswas-speaking Yupi tribe and the Amamutsin tribal band. And here's the official UCSC land acknowledgement.
We would also like to thank our generous sponsors, the Santa Clara Valley CNPS chapter, UCSC's Plant Science Fund, and the Norris Center. There were so many individuals who made this project possible and supported our research along the way. We would like to thank all our friends and family for supporting us over the course of our theses. So why restore ecosystems? The UN has declared 2021 to 2030 the decade of restoration. And if we are going to meet the 30 by 30 initiative, which outlines the need to preserve 30% of the Earth's surface by 2030, we are going to have to do a lot more ecological restoration. Restoration aids in the recovery process in an ecosystem that has faced some form of disturbance. In the modern age, ecological restoration is important to mitigate the effects of climate change. Restoration can also help reestablish ecosystem services. An example of this would be in California grassland ecosystems, where restoration can increase carbon storage, reduce soil erosion, and increase water quality.
So in restoration ecology, there are two main revegetation techniques that are used to increase native cover, direct seeding and outplanting. Seeding generally consists of broadcasting seeds over a wide area, and it's generally lower in cost, but it also has lower rates of plant establishment. Outplanting, on the other hand, consists of germinating soils, like in a greenhouse setting, prior to planting out in the field. Planting is more time-consuming, labor-intensive, and costly, but it generally results in greater plant establishment. So restoration compliance goals are frequently evaluated through current standing native plant cover, but this doesn't always provide an accurate picture of what species are present at the seed bank. Seed banks have complex dormancy mechanisms, which are really difficult to quantify out in the field. Additionally, seed banks are really important for Mediterranean type climates because seeds can persist in the seed bank for many years until favorable conditions occur. They're also important for the long-term restoration success and sustainability by contributing to a self-sustaining ecosystem, which is obviously the main goal of restoration. By evaluating how differing seeding and planting revegetation techniques influence the site seed bank, we can inform practitioners about how to better achieve restoration outcomes and goals. Seed banks are very important for restoration longevity and therefore the overall success. So it's really important to take seed banks into account when considering compliance monitoring. Generally, there's a few limitations to compliance monitoring due to like limited funding, lack of oversight, and compliance standards are occasionally expecting more than what practitioners can realistically provide on a site. And restoration outcomes are generally measured through the percent vegetation cover, but this doesn't really take into account pre-restoration conditions or regional species distribution or historic disturbance regimes in ecosystems. And lastly, it's also a lot harder to quantify species richness over a large area versus like a smaller restoration plot.
So I just wanted to broadly cover nitrogen deposition before we get further into our talk. So NOx emissions and nitrogen deposition are increasing on a global scale as a result of anthropogenic emissions from large vehicle tailpipes and overuse of synthetic N-based fertilizers in agriculture. Nitrogen deposition results in nutrient enrichment typically favored by non-native species, which can alter what species germinate from a site seed bank in the long term. This nutrient is more efficiently uptaken by faster growing non-native species that are better adapted to high nutrient soils. As a result, there exists a competitive advantage that increases non-native species' chance of survival over native species when nitrogen is present. Knowing how simulated nitrogen deposition can affect what seedlings germinate from a site seed bank can be useful supplemental knowledge to restoration practitioners, as California grasslands already have many non-native species. Fertilizer concentrations for N-treated flats in this experiment were made to emulate the rates of dry nitrogen deposition in the Western United States. This ranges from 3 to 8 kilograms per hectare per year.
So with my research, I hope to address several specific questions over the duration of the project. My first main question was, will projects restored through direct seeding methods have a greater relative abundance of native species than sites that were restored through outplanting methods? Another question I had was, will projects restored through differing revegetation methods have different leaf trait values? And my last question was, how will the percent ratio of native and non-native species observed in my seed bank samples compare to the vegetation surveys conducted in the field? And I hypothesized that sites restored through direct seeding methods would result in a more comprehensive species composition than planted sites. I also expected to see a lower specific leaf area, lower leaf lobeness, and higher carbon to nitrogen ratio and greater delta carbon 13 in planted sites than seeded. And lastly, I expected seed bank samples to be highly dissimilar to the vegetation surveys conducted at the time of their collection.
So I posed the research question, Does nitrogen addition affect the abundance of native and non-native seedlings germinating from the seed banks of restored California coastal grasslands? I expected to see more individuals for non-native species than native species, given that most California grasslands are dominated by non-native species. Nitrogen has the ability to increase the fertility of the seed bank, which is believed to benefit faster growing non-native species over native species. So the soils we used for our experiment were collected from 8 California coastal grasslands that were restored using either direct planting or seeding methods. We obtained our sample soils from different sites that range from Humboldt County to Santa Barbara counties. Our samples were obtained from the top 5 centimeters of soil after the removal of standing vegetation, and each site had 3 to 11 transects where soil was collected with samples taken from each transect. Transect sample soils were homogenized in the lab for each site for use in this study. The sites on the left were restored through planting methods, while the ones on the right were restored through seeding methods. You can see the counties next to each site.
So a little bit about our soil collection and germination methods. As Alyssa said, we selected 8 sites, and out of these sites, 4 of which were planted and 4 of which were seeded. And we paired these sites geographically. So we had 2 sites, one that was seeded and one that was planted in the same general region with a similar climate to make analysis a little easier. We germinated the soils in 10 by 15 centimeter seedling trays, and we layered 150 milliliters of collected soils on top of a layer of like just sterilized greenhouse soil. And we decided to do this because if we just did all collected soils, we wouldn't have enough for all of our replicates. And B, we determined that like if the seedling tray was just filled up with the collected soils, then it was very unlikely that all of the seeds at the bottom of the tray would germinate, which wouldn't really help with our study. And we had 5 trays for each site. And so we had a total of 40 trays per person. And I compared mine to field surveys, and Alyssa compared hers to the effect of nitrogen. My experimental treatments were seeding and planting. So I compared the relative abundance of individuals from both techniques to see how they differed.
So all of my seedling flats were treated with liquid-based nitrogen fertilizer, and Nick's flats served as my control. I diluted 0.3 grams of ammonium sulfate at 2100 concentration into a 3.8 liter gentle watering can and dispersed them evenly across flats every week, once weekly after watering. When germinations were spotted, they were marked with unique colored toothpicks until positive identification could be made. After positive identification, the seedlings were removed from the flat. The species that we could not identify immediately were transplanted into individual larger growing containers containing ProMix to promote growth. Nick and I use the same methods to count and mark our germinants.
So a little bit more about our data analysis. Once the seedlings germinated, we identified the individuals, and once they grew 2 sets of true leaves, we obtained leaf samples and then counted the individual by removing them from the tray. Once we obtained these leaf samples, they were scanned and analyzed utilizing ImageJ technology. Once we had our data collection all set, we did statistical analysis, which was conducted using the statistical software JUMP version 16. And then the image on the right is a scan of Medicago polymorpha, which is a non-native forb species, which we encountered a lot of, mostly in Watsonville and Glass Beach.
So Nick and I used analysis of variance, or ANOVA tests and t-tests to identify whether our data had statistical significance based on p-values and f-values. To analyze my data, I tested how the treatment nitrogen addition affects 4 guilds of germinants. Guilds were classified by plant locality, being native or non-native, and growth form, graminoids or forbs. The growth form graminoid encompasses grasses, rushes, and sedges. I compared the relative number data from flats that had nitrogen addition to those that did not to try and identify shifts in species abundance between the treatment and the control.
So I measured the leaf lobeness, leaf area, and specific leaf area for all the samples that we took. Leaf lobeness tells us about the potential for a leaf to cool via conduction or convection instead of transpiration. So a greater lobeness means more capacity to cool with surrounding air. And on the slide right now, you can see an example of low leaf lobeness in the native Rumex species that we recorded. And then the non-native Erodium botrytis has a higher leaf lobeness. And then leaf area is really important biologically and is related to a plant's photosynthetic capacity, and greater photosynthesis requires like more nutrients. It's important ecologically because it can make individuals better competitors. California species are generally slower growing and a lot more resource conservative, while on the other hand, native or invasive species are generally the opposite, and they grow really fast and choke out native species. So on the slide now, you can see a low leaf area of native Juncus buffonus and a high leaf area of the non-native Hordeum species. And lastly, specific leaf area, which is a function of leaf area and dry mass, is related to a plant's herbivory resistance, leaf longevity, lifespan, and overall drought tolerance. And on the slide here, you can see an image of a low SLA with Bromus diandrus, and the highest SLA that we recorded was Juncus buffonus. So a little bit about our results. These two figures represent the total abundance of grass and forb germinants as observed in my greenhouse replicates. On the y-axis of these figures is the relative number, which was calculated following data collection using both native and non-native species. The relative abundance of germinated seedlings didn't really differ between seeding and planting treatments of any of the four guilds. The figure on the right shows that both planted and seeded sites had a trend of higher grass germinants, with seeded sites having a little bit higher than planted sites, but these results weren't statistically significant, so they're just trends. That being said, it was consistent with what we anticipated because typically seeding efforts utilize like faster growing annual species, while planting efforts generally focus more on like slower growing perennial forb species. And then for the leaf traits, I found that forb specific leaf area was the only statistically significant trait, with the mean SLA for seeded sites being greater than planted sites. I found that there were no statistically significant grass traits, which was kind of surprising.
So just a little bit on the nitrogen results. So my y-axis in these figures is representative of the relative number I calculated from my removal counts of native and non-native species. Relative number represents the proportion that one growth form occupied for each tray. The figure on the left reports results about the relative number of native and non-native graminoid germinations in nitrogen treatment flats and the control. Nitrogen did not have a significant effect on non-native graminoid germinations. However, it did have a significant effect on native graminoid germinations. This means that nitrogen addition decreases the relative number of native graminoids present in N-treated flats when compared to the control. The results on the right show the relative number of native and non-native forb germinants from the control and nitrogen-treated flats. Nitrogen did not have a significant effect on the relative number of both native or non-native forbs. So one thing that we did observe in our experiment was below ground biodiversity. California grasslands are often dominated by non-native annual grass species. However, we recorded a relatively low number of germinants for this guild. This exhibits below ground biodiversity that exists in seed bank samples. Below ground species composition often resembles above ground composition, but can vary in regions with high climate variability, such as California. We recorded many non-native species, particularly forbs, but we did record some natives, including Juncus buffonus, Lupinus bicolor, and Aquispawn parviflora, despite our expectation that we would see mostly non-native species germinate. One of the reasons we may not have recorded as many grasses as previous studies is because of how our soils were treated prior to the study. After collection, soils were heat treated and put in storage for 3 years, which may have reduced the number of viable grass seeds that could germinate.
So as I presented in the figures, I found that revegetation method didn't really have a significant effect on overall species germination rates. While there was a slight trend in seeding being more effective for establishing grass species and planting being more effective for orb establishment, they weren't statistically significant, but it is consistent with current practices. The results for leaf traits may be largely due to the types of species that are selected for each method. As I mentioned, seeding generally favors faster growing annual species, and planting usually utilizes a combination or utilizes more slower growing perennial species. Future restoration efforts should focus on utilizing a combination of both methods to better replicate like pre-disturbance community composition. Additionally, restoration projects, when possible, ideally would resemble or consider the type of ecosystem that they're attempting to restore and kind of try to tailor the restoration techniques to that ecosystem if possible. One thing that I found while I was doing this research was that it's been documented that seeding efforts are generally more successful at reestablishing seed banks for sites that have like less disturbance adapted seed banks with like more like naturally transient. Functional traits of native communities can also be good indicators of a community's resistance to invaders. A practical implication for this would be for managers to select native species for restoration that have comparable functional traits to the invaders, which would better prepare the ecosystem for like future changing conditions. And while the trends I observed in the data were consistent with what we were expecting and previous studies had found, these trends were largely driven by native species that were not reintroduced during restoration, which was really surprising to us. There were species that we recorded below ground that weren't planted, mostly Juncus buffonus, which was driving these trends. There were also a lot of species recorded in the vegetation surveys that didn't germinate in our flats, which, as Alyssa was mentioning, it may have been just sort of like some of the conditions that happened prior to this study. But it also may suggest that reintroduced species may not be contributing to site seed banks as effectively or in the way that we're like would be hoping.
So just some nitrogen experiment take-home points. I was able to identify that native graminoids are particularly vulnerable to increased nitrogen deposition. Increases in nitrogen in soil can decrease the number of native graminoid seedlings that germinate from the seed banks of these 8 restored sites. In the long term, this may require maintenance as anthropogenic NOx emissions continue to increase and result in higher concentrations in soils. With future efforts to preserve these sites, practitioners should focus on how they can better introduce native graminoids into sites with high nitrogen concentrations or nutrient enrichment. This can be done by introducing native species that are better at uptaking high amounts of nutrients without experiencing toxic symptoms, such as lupins. It is often difficult and expensive to ameliorate soil conditions. Therefore, point sources of pollution should be identified and managed. Legislation should aim to create better small quality standards for larger vehicles to decrease the amount of NOx emissions that are emitted by each.
So once again, we wanted to thank all of you guys so much for coming out to support us tonight. And thank you again to CNPS for supporting our research. We really appreciate all the support that we've received over the course of this project. And we welcome any questions or comments that any of you have at this time.
Thanks very much to both of you. We do have some questions. That was a really, really well-practiced, very good presentation, and thank you for that. First of all, who did the restoration sites? Were they by the same organization?
There were, I believe, 35 restoration sites that were surveyed, and they were all like done by different organizations. That our project kind of piggybacks off a previous project that our mentor did. So we used the collected soils from his study.
Okay. So this question kind of follows up on that. Did you figure out the type of soil for each restoration site? I'm wondering if a sandy soil would not be impacted by additional nitrogen.
That wasn't really something that we studied like during our experiment, but it was kind of interesting to see how the soils like differed from each site. Alyssa and I, we were kind of commenting on it when we first started because we like had all our soils laid out in the greenhouse. And whenever we would water them, they would all like interact a little differently. Like some of them, they all like retained their like hydrology from the field, which was really cool to see.
Mm-hmm. Mm-hmm. How did you determine the best location for collecting soil for each site? Know that was predetermined already, you said.
So we picked 4 planted and 4 seeded sites. And like we mentioned in our presentation, we paired planted and seeded sites geographically. So we would have a similar climate for sampled sites.
Mm-hmm.
We decided which sites to use based on advising from our advisors who are both here this evening. So thank you so much for helping us along the way. But we did have 8 sites and they did range all along the California coastline.
Great. I see a response from Justin, who I guess is one of your advisors, saying none of the soils they used were sandy texture, mostly heavy clay.
Mm-hmm.
So good. And were there any confounding variables that you both focused on controlling or accounting for? I think that's the last question.
There weren't any that we kind of were addressing like during the experiment. After we kind of like considered how like the like previous handling of the soils could have impacted our results, that like Alyssa kind of talked about that a little bit, which we couldn't really like mitigate for at all, which was kind of unfortunate, but yeah.
And one other question popped up on YouTube. Did you look at any mycorrhizal relationships?
No, that was a little out of the scope of this study, but maybe in the future. That would be very interesting to look at.
Master's thesis project, maybe.
Yeah.
All right. Does anyone see any other questions? I think that's it for now, but terrific job, you guys, and great job. Answering questions is hard, so thanks for that.
Thank you all so much. We really appreciate this opportunity.
Okay, then our next presenter is Reid Kenny. He is going to be talking about his project, which is systematics of the genus Juncus using DNA analysis of various gene regions. Reid is currently in his 2nd year as a PhD candidate at UC Davis. He's going to be sharing his preliminary work on his dissertation with us tonight. He has a background in field botany and California floristics, and he hopes to someday teach botany as well as to conduct research in California. So Reid, go ahead, please. Thank you.
Hi, thanks, Vivian. And just wanted to say what a pleasure it is to watch Nicholas and Alyssa. I did my undergrad at UC Santa Cruz as well, so it's good to hear what's going on there. Let's see, I will share my screen. Okay, so yeah, I will be talking to you all about systematics, actually in Juncus in general, as we will be talking about several genera in the Juncus, although my true passion is for the genus Juncus. And before I get going, I will also go ahead and recognize that my university is also sited on the land of indigenous Californian peoples, the Patwin tribe, and here is our university and tribally sponsored land acknowledgement statement. I also have conducted fieldwork all over California, so we'll just acknowledge all of the various indigenous peoples that have and do occupy that land. So, a little order of events here. I will just talk briefly about taxonomy in general, why we should care, do a little bit of jargon busting. Hopefully, I'm going to try not to use very many technical terms in this presentation, but there's a couple things that are going to be useful. And just give a really brief overview of what we know about relationships in the Juncus. Then I will talk about my methods. I'm going to leave out the laboratory methods, but I just wanted to take you all with me on a photo journey of some of my collections over the past year, looking for Juncus in California. Then I will show you what I found. So preliminary results of my work, and then I'll just briefly touch on what I'm hoping to do in the next few years during my PhD program. So why should we care about taxonomy above the species level? I assume that folks who attend CNPS meetings care about the identity of the plants that we all love to look at so much. But I know that it can sometimes be frustrating when what family a particular genus is in has changed, several times in the last 10 years, or you keep having to learn new genus names for the species that you like to look at. I feel that pain as well, but I just wanted to go over a couple of the important benefits of this worldview that puts forward that it's useful for our taxonomic names to reflect evolutionary relationships, and those relationships are still being uncovered in many ways. So a few reasons why that's particularly useful. One, and this is a very basic reason, but this is a very, it's very important for a lot of science in terms of anything that compares many species, maybe across the globe. It's really useful to know what characteristics of a species might be shared because they share common ancestry versus what characteristics may be favored in certain ecosystems. So questions like that, it's really important to actually know those evolutionary relationships. But aside from sort of theoretical research, there's a lot of really practical reasons why it's very useful to know these evolutionary relationships. So if you think about invasive species, some of you out there might know, and Alyssa and Nick mentioned some invasive species in their work. For example, there are many species of bromus that are invasive in California and North America in general. And in fact, when you're looking at analyses of how to predict what species across the globe may be invasive in countries where they have not already invaded yet, their evolutionary sort of closest relatives and whether those species are invasive is a big predictive factor. So if we know that one bromus is invasive, we might then also have information about the tendencies of other species in that genus bromus. And for a Juncus example, Juncus effusus is actually non-native. It's not an aggressive invader, but it's non-native in much of its range. And there are a ton of species that are closely related to Juncus effusus in Australia. And so who is to say which one of those species may or may not become the next bad weed choking up the California Delta? And this is why it may be very useful to know what those species are related to. But also things like therapeutic compounds. If we find some chemicals in one plant that we may think have some really useful benefits for human beings, it's very likely and very often that close relatives of that plant may produce similar chemicals with the same or closely related beneficial functions, and they may also be more available, say, or more useful for extraction what have you. There's also big benefits in the cultivation and horticultural space as well. If you are trying to figure out how do I grow a plant, one of the first things you might be interested in say, well, are there any other plants that are already in the horticultural trade, in cultivation? What are their requirements? And that's going to maybe be my starting point for bringing, say, a new species into the trade. And if any of you grow plants in the Proteaceae, say, that whole family tends to be very sensitive to phosphate fertilizer. That's a characteristic that is conserved in that family. And so knowing that gives you a lot of information about, well, if you're trying to grow a plant in the Proteaceae, then you're definitely going to need to be careful with the fertilization regime. And then the other big point I just wanted to say is that what a species is potentially able to cross with is also really important and informed by these evolutionary studies, such that many people work on wild crop relatives. A lot of people in my lab are working on these questions of how do we identify what wild species are the closest relatives of cultivated crops so that we can start breeding programs to get those beneficial genes and diversity back into our crops. So that's all just to say that, taxonomy and systematics is not just for basic research purposes all the time. Okay, so just a couple terms that you may or may not be familiar with, but that I find useful to use. Just the first one is this word phylogeny. All this means is a family tree of species, so similar to a family tree of people, although we like to draw them so that they're only bifurcating, say that they're a little different than family trees of people, but you can think of them as representing species and their relationships to each other. And that word phylogeny just means this diagram here. And then the word clade means a group of species that all share a closest common ancestor. So here in the red box, C and D are species that share the closest common ancestor E, and we would refer to that as a clade. So what is up with the Juncaceae? The big factor here that drew my attention initially a couple of years ago when I was thinking about what I wanted to work on in my PhD is the fact that within the Juncaceae, there you have two large genera, Lugula, which some of you may be familiar with. A lot of them look kind of like Juncus with hairy armpits, so they have these hairs on the sort of axils of their leaves. And then you have Juncus, which is what I was originally interested in studying. And the work coming out in the early 2000s, early to mid 2000s, showed that Juncus, some species in Juncus are actually more closely related to several genera in Juncaceae of these South American cushion plants than they are to the rest of Juncus. So that's illustrated here in these lower nodes of the phylogeny. And that set up this question of sort of this taxonomic problem. Okay, is this actually the case? If so, do we need to change our taxonomy? And does this have implications for how we understand sort of the organization of the genus and the family? So that was the starting point for my research. And then really wanted to dig into what's going on with these Juncus species that are appearing to be closely related to the South American Cushing plants. So those Juncus are two groups. They've been named as sections within the genus Juncus. So one group, one section here has been named Cespitosae. They are extremely small annual Juncus. So scale bar here, about 1 centimeter for this drawing, and we'll have some pictures of those coming. And then the other group are these grass leaf Juncus. So these are Juncus that don't have the typical round leaved morphology. They actually have leaves that look very similar to grass, although they have flowers that match typical Juncus morphology. So those are the two groups that I set out and I'm still working on resolving the evolutionary relationships within these groups, between these groups, and between these groups and the South American cushion plants. So that, broadly is what I set out to do. In order to achieve that, I had to collect some Juncus from all over California. So we have many representatives of those sections Cespitosi and Graminifoli native here that had not yet been sequenced for their DNA and represented in any phylogenies. So that has been my first goal for the first few years of my PhD here. So last summer of 2021, I set off on some road trips in California. So wanted to share with you guys some of my adventures and misadventures. So my first destination in late May of 2021 was from Davis. I went up towards Lassen National Park area, had heard reports of a lot of annual Juncus up there. Got up there and this is what it looked like. So it was quite chilly. There were not a lot of plants up yet, which is always the first big challenge when you're going to look for plants, is trying to figure out what the weather and the phenology is going to give you, especially when you're dealing with drought years. So I was pleased that I was not too early, and I did do some scouting around on that trip, and I did find some annual Juncus. Although to my chagrin, it turned out to be Juncus bifonius, which we've been hearing a little bit about. Although a lovely species, not one of the ones that I was hoping to sample, but had a beautiful trip nonetheless. And the next week, tried again a little more locally, headed over to Lake Oroville, and got my first collection of that season. This is Juncus capitatus, actually not native in California. It's of European origin, widespread invader of many habitats, although it's so diminutive that I doubt it has any real ecological impact. And so there it is with a millimeter scale ruler to give you an idea of what we're dealing with here for trying to find and collect this section Cespitosi in general. Was really felt like my big challenge for the summer, is how do I track down these tiny plants so that I can sequence them. But with that little bit of success under my belt, I headed down to San Bernardino for my biggest collection trip, down to Big Bear Valley, and some beautiful landscapes down there that I really enjoyed getting to explore and collect in. So this is the sort of habitat where you find a lot of these annual Juncus. They're sort of on creek edges or in vernally drying sandy flats. Here is one of my target species there. That's Juncus teamii. And just to prove that I was really there, here's me. I do not have a 4-wheel drive vehicle, so I had to use my mountain bike to get around to some of these collection locations. And I also made it up to Lake Davis to collect the rare Juncus luciensis, which was exciting. Redocumented a couple populations there and was given permission to collect them for my work. And then finished up my season with several trips up to Lake Tahoe late in the summer, Donner Pass area, to get these high elevation species that tend to grow in these sort of granitic sands that dry late in the year. So there's Juncus capillaris. And those have all been those members of section Cespitosi, these really small annuals, which I was not surprised that people had not done much sequencing of yet, because they are quite hard to track down. And here's another photo to give you a sense of what the survey process tends to look like for finding these plants. Kind of walk 10 steps and then get down on your belly and see if you could find any. Okay, so I collected all the plants I was looking for. I did a bunch of lab work, and I'm still doing lab work to extract the DNA, amplify it, and sequence the gene regions that I'm interested in, but I have sequenced the internal transcribed spacer region of several of these species and have some preliminary results. So I didn't want to put up a bunch of these phylogenies because I know they're kind of a pain to look at for most of us. But here, this is the sort of the important basis of my results here, which is that you can see this clade of South American cushion plants coming out here. And here we have all of our related Juncus species. So in North America, like I said, we have these grass-leaved Juncus. And here we have our little clade of annual Juncus as well. And so there's a few interesting things that we see here. We see one, that the North American annuals and the North American grass-leaf Juncus appear to form a clade, appear to be closely related to each other, and then also appear to be closely related to the South American cushion plants. And here at the top of the diagram, there are several of these South African species of the grass-leaf Juncus that look to actually be less closely related to any of the rest of the species. So that indicates that there are several things about the groupings of these species that we thought were true that aren't true, and that are really exciting and have some interesting biogeographic implications. So my future goals for this are to add many more species, hopefully everything in those sections, Cespitosi and Graminifoli, as well as some more of the South American cushion plant and related species and genera, and really unravel the story of what are the relationships sort of in this larger group, and what implications do those relationships have for taxonomy and systematics in Juncaceae as a whole. So yeah, and so that's sort of the phylogenetics side. There are a few other aspects to this work that I'm really interested in. One is the sort of broad scale biogeographic implications, in that we have these species in South America, South Africa, and North America, and we have methods where we can sort of try and trace back where was the origin of these groups and how did species migrate between continents and radiate and, you know, turn from something that looks a lot like a Juncus into a wild high elevation cushion plant, or become annual and have species become annual in multiple events. That sort of question we can begin to ask. There are also some interesting ecological aspects of Juncus. This is a couple photos of scanning electron microscope photos, actually, of Juncus seeds, of which there's been a lot of documentation for phylogenetic purposes. But now that we have molecular evidence for phylogenetic work, we may be able to use some of the data collected on these seed coat characteristic to ask ecological questions. In particular, many of these seed coats become highly mucilaginous when wet, which allows them to stick to substrates, may enhance protection. They may be more likely to stick to the foot of a migrating water bird, or perhaps pass through the gut without being damaged. But this trait varies in species. So one question we might ask is there a relationship between seed coat characteristics and perhaps how widely distributed a species is, or whether it follows water bird migratory pathways. So those are some of the things that I'm interested in also pursuing once we have a better understanding of the phylogenetic relationships in this group. And with that, I will wrap up and say thank you so much to you all, the Santa Clara Valley chapter of CNPS, also the Davis Botanical Society, as well as the statewide CNPS for generous funding, also my PI, Dan Potter, and my wonderful lab group at UC Davis. And I would be happy to take questions.
Great, and we do have a few questions. So thank you very much for that, Reid. Appreciate it. It's fascinating how much work you're doing. And are you traveling this summer? Are you going this summer to do more collections, or is your field work kind of done?
Yeah, it's definitely not done, but I don't have any collections lined up for this summer. A lot of the material I'm hoping to get for the future is actually international. So likely will be sourced from herbarium specimens more so than I have been, although I would love to travel to South America and South Africa if I get the chance. But I'm also preparing for my qualifying exam in the fall. So I'll be home doing lab work and studying for more this summer. This summer.
Right, nose to the grindstone. So how did you know which areas to go to in California to collect the plants you did here?
Yeah, that's a great question. There are a few, we are really lucky in California because we have a well-documented flora. So my main resources for identifying good potential areas are herbarium specimens, so actual records of where people have collected these species in the past, and we have really well organized, easy to access database of herbarium collections. It's CCH2, the California Consortium of Herbaria, is the database, and the website is CCH2. So that's that really easily searchable for where to find plants. And I've done this for a ton of species over my career. So that's always go to. And then CalFlora, if you all are familiar with that website, is also just a really great resource, and actually also does draw from herbarium collections as well, but what is a little more broad with field observations included. I also will use iNaturalist when I'm scouting, because I generally can tell from a photo whether something is what I'm interested in. So yeah, those are my three big sources, I would say. Sometimes I'll have a friend who has actually seen something somewhere, which is like the best case, but it is always nerve-wracking to roll up to a new place. And yeah, that's where the rubber hits the road. A herbarium specimen will get you in a quarter mile, but then it's just leg work.
Definitely. And maybe a snow shovel too.
Yeah.
So how many Juncus species were you able to collect so far?
So far, I've collected 12 species, I believe. That phylogeny I showed, I had successfully sequenced 7 of them. So that is also what I'll be working on this summer, is just getting additional sequence from those species I haven't worked with yet.
So a couple quick questions on the South American cushion plant, whether it looks similar or resembles the Juncus, and do they grow in or near water like the Juncus as well?
Yeah, they do. Yeah, I should have put some pictures of them in. They grow in these alpine, sort of boggy, seepy areas. Yeah, with perennial water sources, but they look actually quite different from Juncus. They're extremely mound forming, and so they don't have any of the long stems of Juncus, and they have a lot of them have these two ranked, very short leaves. The flowers are relatively similar, but they have some interesting morphological variations. A lot of them have these long sort of protrusions on the female flowers, which actually lift the female flowers. Actually, sorry, it's the male flowers that have the protrusions, and they stick up above the cushion, and then the female flowers are nested within the cushion. And that's another big difference, is that some of the species are dioecious, meaning they have male and female plants, which is not known in the rest of the family. So yeah, there's some pretty serious differences, which is why they were originally named as these separate genera, and why it's so sort of surprising to see them as being closely related to Juncus.
So last question, I think you sort of alluded to this, but there are some rare species of Juncus in California, and I assume as you go through and do all your genetic research, that should inform rarity listings and ecology, or how do you see that? Do you see any of those maybe changing, or some clarification?
Yeah, that's a great question. It's actually the level, there's some possibility for that. So the Juncus luciensis that I collected was from the Northern Sierra Nevada area, Northern Sierra Nevada Cascade ranges. Juncus luciensis also occurs in the Santa Lucia range, so down in Santa Cruz and Monterey, and it's rather disjunct. So I wouldn't be surprising if there actually is some genetic separation that has happened there, but it also depends how recently that population sort of divergence has been established. So it is something I do want to do is sample the Juncus luciensis from the coast, and was sort of toying with looking at that as a potential project. In general, though, my work is at a broader scale than would inform something like rarity rankings, where we would tend to collect more population level data and ask questions like I was just saying, are these maybe two just disjunct occurrences actually a single species, or are these less related to each other than we might expect from morphology? And currently, I don't have plans to do that kind of work. But of course, if there are funding opportunities that come my way, I would be happy to.
Definitely. Well, thank you. I think that's it. If anybody else, anything else, put it in the chat. But we really appreciate you talking about this, because I think a lot of us amateurs kind of overlook the Juncus, the whole family, as much. And it's nice to see appreciation and work that you're doing on this. So thank you very much for your talk.
Yeah, my pleasure. And I hope to be representing Juncus in California for a while to come.
Great. Send all your questions to Reid then. All right.
Thanks, Reid. That was great. We also appreciate your wall art too. Okay, so our next speaker is Lauren Hamm from UC Berkeley, and her project is the evolutionary dynamics of climate stress responsive mobile elements in the yellow monkey flower. And she's with the Blackman Lab at UC Berkeley. She's only been in California for 3 years, but she's already fallen in love with the flora here. So Lauren, it is yours.
Hello. I'm excited to be here. Let me go ahead and share my screen with everyone. All right, I think that should be it. So I'm going to go ahead and get started because it's late and I'm sure everyone is very tired. But if you can hold on just a little bit, I have an exciting story to tell you about, in particular, how Mimulus guttatus fits into how we can better understand plant responses to climate change. And coming from the wake of Reid's presentation, I do want to go ahead and clarify that for those of you who do pay attention to recent name changes, Mimulus guttatus is often also called Euranthera guttata. For the purpose of this talk, and because a lot of the literature still refers to it by its older name, I will continue to call it Mimulus guttatus, but I did want to go ahead and say that there has been a name change. So if you're worried about it, you are right. That is actually what it is called. So I think I'm going to go ahead and start with the big picture, which is climate change, something that we're all very aware of that's happening. And I sort of wanted to zero in on two parts of it, which is first, on the left here, you can see projections over the next 50 years of an increase in temperature over different sections of California. And what you can see is that overall, over this period of time, we expect to see about 2 degrees increase in Fahrenheit in temperature over this time. It does vary a little bit, but the overall trends are quite clear. If you think it's hot now, it's going to get hotter. But not only will it just get hotter on a global scale, what's really particularly important is that we see these big changes in extremes, oscillating extremes. So we'll have really high temperatures and we'll have really low temperatures that are unexpected and often unpredictable. And that's sort of visualized here by a paper that came out a few years ago. On the right-hand side, if you follow that sort of red line, you can see that there are these record-breaking monthly temperatures, both in the positive and negative aspects, so colder than we think or hotter than we think, and that they're already happening way more often than what we would expect. And this is definitely our fault. Not only are these changes and oscillations happening with temperature, but they're also happening with precipitation. So what you're looking at here is a percentage of average precipitation over the month in question that you can see, with the top row being the water year 2018 and 19, and the bottom row being the following year. And what's particularly important is on a scale of precipitation, with red being a very small amount, little to nothing, and blue being a heavy amount of precipitation for the month. The month of February over these 2 years it showed an incredible amount of difference between the two. And this is, for California, one of the biggest periods of rain that we're going to see is in this month. And over a period of 1 year, we see a difference between some very, very high amounts of precipitation and then suddenly a drought year. And that's really common for this area. And as someone who hasn't lived here for very long, I've already started to see it. There's incredible amounts of variation on a year-to-year basis that are often unpredictable, which isn't that big of a deal for us. It's hot outside, we can move somewhere else. We don't have enough water, we can go to the grocery store and get something. But for a plant that's sessile and can't move around, this sort of oscillation, unpredictability is a big deal, and something that they need to worry about developing something called plasticity for, where they're able to respond in multiple different ways depending on what the environment gives them to work with. And this happens for all of the plants that we have here, but in particular, there's a very, very heavy emphasis on how this affects the star of the show, Mimulus guttatus, a personal favorite of mine. This is also called the yellow monkey flower. You've probably seen it all over the place. It grows just about everywhere, which I'll get into in a little bit. It's called yellow monkey flower because it has a cute little flower that supposedly looks like a monkey's face. I'll let you debate whether or not that is true. I just think that they're very cute. But they're not just cute, they're also really ecologically important for our studies because they grow everywhere. And I have put together a sort of a brief slideshow tour of all of the exciting different places that we've collected from, or not all of them, but enough to give you an idea of the breadth of what we have here. So we have Mimulus here and the beautiful little yellow flowers. On the left, this is in the eastern Sierras, growing right along the ridge of a stream. Both of these are high altitude populations, with the one on the left being in the Sierras and the one on the right being at the top of Mount Tam, which is very close to where I live. What's exciting about the Mountown population is, if you know anything about geology, there are these huge veins of serpentine, which is a soil that has a really, really difficult heavy metal ratio and nutrient ratio, in which there's a sort of an offset of magnesium and calcium that makes it almost impossible for anything to grow there unless you're mimulus. One of my lab mates likes to call them the honey badger of plants, because they can take anything. And at this point, I'm starting to believe it. So we do see lots of variation here. But if you look at the lower elevation populations as well, there's still quite a bit of variation. So on the right here, this is in Dardanelle, there's sort of wide open field, lots of competitors, lots of tall grasses. It's a bit harder to see, but if you look closely, there's these little yellow specks. Those are mimulus. They are growing here and they're doing quite well. Drastically different to what you see on the right here, which is Point Reyes. This is near Almera Falls. Growing right on the edge of the waterfall. You can see the ocean in the background. They also grow right up to the edge. Anywhere they can, they'll be. And then this is another good explanation of some differences that we see. On the right here in Prather, we have a population that spans around this river, so much so that it will grow right smack dab in the middle. They like to have their roots sort of like hanging in the flowing water. But Copperopolis on the other side, this is another serpentine site. This is a copper mine tailing. This is an old abandoned mining town. And in this sort of dry, deserted area, you see all these outcroppings of mimulus, these beautiful yellow flowers growing amongst the really harsh metal deposits here, and honestly flourishing. They're doing quite well. So we see mimulus pretty much everywhere. And that gives us a really wonderful system to ask questions about how this plant responds to differences in the environment, because it is predisposed to do well in these really harsh and difficult places. So if you're wondering, like, oh, what does that look like on a map? You can see that here on the right. The general range of Mimulus guttatus is quite extensive. Not only does it cover the sort of entire western part of the United States, but it fills that almost the entirety of the western coast for the entirety of North America. So we see a span from Canada all the way down into Mexico, and even a bit more eastern. We have populations that were sampled off the cliff face of El Cap in Yosemite. We have populations that are growing in national parks amongst these really, really like sulfuric water deposits. What's important is that there's water. That's what Mimulus is really looking for. And if that's there, then it can grow. Some more fun facts about the species before we keep going on. It has an incredibly high effective population size, which means it's very tractable for the kinds of selection studies that I'm looking for, and for looking at sweeps to see if something was selected for and might be potentially adaptive over time. It's also one of the most genetically diverse plant species that's been studied to date, period. And that is great, because what we're looking for is small differences that might make them more plastic and more easily respondable to a changing environment. And then this last bit, which I've alluded to a bit before, their growing season ends in a terminal drought. So the one consistency across all those pictures that I just showed you is that there's water. There's water somewhere, either seeping up from the bottom or flowing nearby. Mimulus is entirely dependent on water, so much so that when we grow them in the greenhouses, we have them sitting in standing water, and they seem to love it. That's what they want most. So if you remember earlier when I was showing you that graph of the amount of water that we see varying from one February to the next February, that's a big deal for Mimulus. If suddenly their entire growing period is drastically shorter, that has huge implications on whether or not this population can survive and move forward. And so Mimulus, like many other plants, has developed this plasticity to respond to these oscillating environments. And so that's why I'm using this species to answer these overarching questions of how climate change might in fact impact these species. Now, often when people look at this, they look specifically at SNPs, or single nucleotide polymorphisms. These are really small mutations that happen in one particular spot. And they often overlook a really important and substantial area variation, which is transposable elements, or TEs. So transposable elements are affectionately known as jumping genes because they jump from one place in the genome to another, either by cutting themselves out and moving the DNA somewhere else to be inserted, or by copying themselves to an RNA intermediate and then reverse transcribing themselves and inserting in the genome elsewhere. So this is a way for a specific gene, regardless of what the rest of the organism is doing, to replicate itself quite quickly. And what's particularly exciting about this is not only does this gene jump around and move, but it affects everything that it lands next to. And this can lead to a bunch of different things. So it can lead to structural changes where, since transposable elements have lots of repeated regions, it can lead to things like inversions or exon shuffling, which means that you're taking chunks of different genes and putting them together to make a new gene that can result. Or you can have something as simple as a transcriptional change, meaning that when a TE lands in a particular region, it can keep a gene from being transcribed and then translated into protein downstream, or it can make it happen at a higher rate, which then changes the phenotype or the important downstream effect of the gene's purpose. So transposable elements are all over. In fact, they are very, very common, and the only reason we haven't looked at them before is because they have these repeats and they're very difficult to look at using computers because of issues that happens with sequencing. In fact, they're so common that even us as humans, half of our genome is consistent not of genes, but of transposable elements. And you might think, well, that's a crazy statistic. If we have all these genes jumping and moving around, wouldn't that lead to genome instability and all kinds of other problems? And that would be a great question, and you would be right. So to prevent that from happening, organisms have developed mechanisms to keep these TEs sort of turned off so that they're not transcriptionally active, they're not jumping around and messing things up, because it's a lot easier to break something than make it better. So the organism decides just to shut them all off. However, there are lots of scenarios in which transposable elements can be turned back on, one of those being stress, something like not having water or it being particularly hot. There's whole suites of transposable elements that are specifically turned on by these stressors. There are lots of others, which I'm happy to talk about. For the purposes of this talk, I'm talking about both heat and drought stress. So there are TEs that get turned on by this, jump around, and then often insert into environmentally responsive genes specifically. So if you put all of that together, it creates this really elegant pathway for a environmental situation to influence directed mutation where it might be needed most. So if you look here in the top right, or at the very top, you have a TE that's turned off, it's silenced. Something happens in the environment that turns it on, activates that TE. It mobilizes and moves around, jumping preferentially into these environmentally responsive genes, and that might eventually result in a major effect mutation. Like I mentioned earlier, a lot of the problems that come along with active TEs is that they mess things up where they land. This whole concept is off of the probability that despite all of these problems, as long as it's not too active, eventually within there, you're going to have something that might be positive and might be helpful, particularly if you're focusing on these environmentally responsive genes and making small changes nearby. So this creates a really elegant pathway for how transposable elements may be a method that allows plants to see how their environment is influencing them and then change how they respond specifically to it. And so I'm looking at this in Mimulus, which we know. This gives you an idea of what that data set looks like. So all the pictures that I showed you before, those are from my lab and other collaborators. We went out by hand and collected all of these plants. Fortunately, it's not at the millimeter scale. They're quite large, they're bright yellow and easy-ish to see from a distance. So when we get into that smaller range, it's decently easy enough to find them. You just have to go out and look. And so the total amount that I'm looking at here, at my last count was, I have 113 different populations of Mimulus that exist along this variation in environments through the entirety of California, and then sort of seeping into Oregon as well. So on the right here, you can see these populations colored by the average temperature for the year of the region, with yellow being the colder temperatures and red being the hotter. And you can see that there's quite an expansive range of these temperatures. And that in particular, when you compare them to the map on the right, which is looking at the average amount of precipitation, whether that be rainfall or snow, that the lowest and highest temperatures don't necessarily correlate with the lowest and highest precipitation. So because of that, we're able to look at all these populations and really tease apart the two and say, is heat what's causing this, or is drought what's causing this? And what I'm doing is taking this massive section of populations, multiple individuals from each of them, and doing gene environment analysis and seeing what transposable elements are moving around and if that correlates with a particular stressor, either heat or drought or both. And that will give us a better idea of what's really going on in this system, and can, for the first time in an outcrossing species, can we show that this is happening and is being potentially adaptive? And this will give us a better understanding of mechanisms that are available to plants to help them respond to these environments. And in particular, although we have a very good indication that this is true, it will be solid proof showing that this is something that can be passed down across multiple generations, giving Lamarck another day in the limelight, which I think is very exciting. So unfortunately, at this moment, I don't have those results back because this takes a significant amount of time to do. But I wanted to let you guys know that that's the plan and that's where we're heading. And in the meantime, all of this data is not only being used for my thesis work, but is also contributing to the California Conservation Genomics Project, or the CCGP. Which in its own words aims to produce the most comprehensive multi-species genomic data set ever assembled to help manage and protect regional biodiversity in the face of climate change. And what that means in simpler terms is that they're taking, they're working with, I think, over 100 different researchers to get, based off of the last count I saw, 236 different species, including both Mimulus guttatus and Mimulus laciniatus from my lab, as well as anything from insects to fish to cougars to just anything that lives in California and is a native species. Their goal was to get it included in this project. And so that sequencing is now publicly available for everyone to use and is being published and annotated by a suite of bioinformaticians that work with this project. And they're also working directly with lab management to try to use this resource as a way to better protect the diversity of the native plants that are here in California. So if you're not already a part of this and you're interested, give them a look. Got to give them a shout out. They were super helpful in getting the work done for this, and I'm very proud of contributing to this resource. So that gives you an idea of sort of what I have right now, where I'm going with it, and some adjacent work that it's also being used for. And then with that, I'm going to go ahead and say thank you to everybody in my lab and everyone who's given support, whether that be financial or emotional or intellectual. Thank you all so much for helping. Yeah, and I'd be happy to answer questions about any of this. I am a big fan. I love to talk about it. So send some questions my way.
Thanks, Lauren. That was great. And lots of nice pictures. And we have some congratulations coming to you from the chat too, and finishing up your year. Mm-hmm. So we actually, as you were talking, some of the questions got answered about these monkey flowers requiring a lot of moisture, mm-hmm. You know, especially in the super dry places. But I think you answered that pretty well. I don't know if you want to elaborate on it.
So a lot of the populations look really dry and are often very dry. And they have seasonal water that comes out. So there's water flows that comes in these seeps that will sort of flow out, and like a lot of them live in very rocky places, and it will just sort of like seep out of the rock. So the water is there, even if you can't see it. The thing is, once the water disappears, the plants only have so much time to reproduce before they start to shrivel up and wither. So that's a huge pressure on the plants, and that's a great question to ask.
Great. So then there was a question about animals having transposable elements, but then you answered that. So another question is, how stable are the transposable elements?
I think that they're largely quite stable. I think I can interpret that question a couple different ways. So they're evolutionarily speaking, they're quite stable. They're 50% of our genome has them, 50% of Mimulus has it. Corn has 85% of its genome as transposable elements. I think there's only a couple species that don't have them, and they're all parasitic and inside of other species, and often rely on what they have as well. So evolutionary, they're quite stable. When it comes to when they're being turned on, and if their activation is stable, that's a bit more variable, because like I mentioned, if you have them jumping around and messing things up, the structural component of the genome kind of gets compromised, and there starts to be all these changes that you don't need. And so because of that, there are some really elaborate overlapping mechanisms that keep them repressed, or after they get turned on, stopping them so that they don't perpetually copy themselves. So they're usually, depending on the different type of transposable element, there's only a certain number of times they can do it before they stop, or they get sort of clustered into these heterochromatic regions, which are areas of the genome that are really tightly packed. So it's hard for the transcription machinery to get in and actually physically interact with the genes. So there's a lot of mechanisms to keep them turned off. And then even after they get inserted into the genome and cause changes, if they're negative changes, they can be taken out of the genome. So through this is on a multi-generation scale, they can be sort of removed from the genome over time. But what's exciting about this, it's called the TE thrust hypothesis, if you're interested in reading more about it, is not to discount them being turned off or silenced often. It's that there's a small number of them that this will not happen to, that might potentially be adaptive and useful, and that we're really interested in finding out what those are, so that we can perhaps sort of put them into other things and see if they can add a bit more plasticity to the genome. Yeah, so I think that answers that.
I have invited the questioner to unmute if she needs more information. Mm-hmm. So, I had a question. Oh, I think your answer was good. Thank you. So, I had a question from Reid. Are all of your collections perennials?
Yeah, so there are actually two ecotypes of Mimulus cutatus. There's perennial and annual, and all of mine are the annual variety. Although there are perennials, we're more interested in the ones that die because of that terminal drought, although there are some that do last over multiple years and will overwinter. Unfortunately for us, the ones that overwinter and last long only do that because they grew in places where they do not have water stress. So they're often found on the coastline where there's constantly water available, and those are the perennials. They also have huge inversions in their genome that make them incompatible to look at in the same experiment without accounting for it. So all of mine are annual, but perennials do exist.
Great answer. So, we had one request to make a t-shirt out of your opening slide, which I don't remember offhand, but we have some t-shirt collectors here. Everybody loves these monkey flowers. There's a note also in the chat that they're always charmed by the variation and differences in the coloration and the flower structures. And I think, as you travel around the state, you see some with red dots and some with, just how they're different. And I always thought eventually it would turn into multiple species or subspecies at some point, but that's not what you're implying here. Is that correct? Or
There are multiple different species of Mimulus that are similar, that are yellow monkey flowers that do have the red dots. Laciniatus is one of those, Nasutus is another. Those vary in size, and they're also selfing as opposed to outcrossing, so they do exist. But there is a lot of substantial variation within just Mimulus cutatus itself, and that's anthocyanin deposition. If you see these in the wild, they actually, the whole plant senesces, and so the leaves and the stem will turn this like deep red purple color as well. I think there's a lot of work being done in my lab and in other labs about the UV patterning. So the differences in the flower that we can't see with our eyes, but that pollinators like bees will be able to see, and then how that affects how the plants grow. And so there's a lot of variation in that might lead to some subspecies, but we're still working on that currently.
A little ways to go. One more question. Are you seeing variation in the plant's appearance due to the TEs?
Not that I've seen so far, but that is really common. A lot of the variegated phenotypes that you see, variegated meaning little tiny spots and dots, like in those species of corn that are sort of very mosaic-y, that is because of transposable elements. The differences in fly eye color, some of the differences in moth wing patterning is that I don't think that's what's happening in this case with Mimulus, but that does definitely exist in the biology kingdom.
Great. I think that's our last question, unless anybody has anything else. So thank you, Lauren. That was terrific. And we really look forward to seeing more of your work. So thanks for coming tonight. We appreciate it.
Yeah, I want to thank all of our presenters tonight. Those were both incredible projects and fantastic presentations. So thank you all for joining us tonight, and thank everybody for coming here and listening to our talks. We will have more scheduled soon. We don't have any on the schedule right at the moment, but stay tuned. Subscribe to our news group if you haven't yet, and you'll get a notification when the next talk is scheduled. And I think that wraps us for tonight. Thank you, everybody. And